everybody. Thank you for coming today to today's Center of Excellence seminar presentation. Today is, as you know, soil genetic testing to assess Fusarium pressure. So we appreciate you coming. I do want to make mention of a few introductions. Sonnet Nelson's in the back of the room. She's the one that made this all happen. Rosa Bevington is here, going to be doing some filming so people can watch it online, and we'll archive it for later. Let's see. We'll do some other introductions in a little while. This is put on by the Yuma Center of Excellence for Desert Agriculture. And for those of you that don't know, we're a public-private partnership. We're funded by the agriculture industry, housed in the University of Arizona College of Agriculture and Life Sciences. So it's we've kind of got one foot in industry and one foot in the university. And that allows us to really make sure we focus on the problems that industry wants solved. And so when we started up about 2.5 years ago, the first problem they told me to work on was Fusarium wilt of lettuce. So we've been doing that. I thought I'd brief you on a few things that we've done on Fusarium, and then we'll get into to the highlight of today's presentation. So, I do want to recognize there's a lot of our donors are in here. My advisory council chairman, Robbie Barkley, is here, and like I say, several other donors, and we appreciate all of you. Donating money, donating information, because I didn't know anything until I hear from them what we need to work on. So as far as Fusarium, back in 2015 we held the first international symposium on Fusarium wilt. We held it here in Yuma, brought together people all over the world that have been working on it, that have been affected by it. And we also had the first field trials that had been held since 2002 to look at new varieties and new crop protection products. Then in 2016, we had a follow-up conference in Salinas, in Monterey actually, and that was to focus on field trials and research and how we move forward with those two areas. And we also had a second year of field trials, and we actually saw some results with some of the varieties, and so that was promising. And we always get these results out, so hopefully they help you when you're making decisions as far as what to plant where. That's all on our website. So this year, we're doing some research on detection. We've got some work going on in greenhouses over in Tucson, looking at both DNA detection and spectral imaging detection to see if we can detect the disease early and detect it remotely. And then we also just got funding for 2 more years of field trials. So we're going to expand the field trials. We appreciate we had some industry come in and tell us additional things we could do in the field trials that'll give us hopefully some good results, at least some new ideas to try. So we'll be doing that for the next 2 years at least. And then we also have some other research projects that are tying into this. Frank Daniese, there he is here from Earth Knowledge. We have a collaboration with Earth Knowledge, which is a big data analytics company, and a USDA national lab to look at how can we get some predictive tools in your hands to help you avoid the triggers that cause things like Fusarium wilt. And then we also got some funding from the Lettuce Research Council, which allowed us to bring information about lettuce diseases, and we've produced some videos, and that's all going to be coming to our website in November to what we call the Learn tab, with all sorts of information about that. So we want to become more and more of a resource for the information you need to fight this disease and others. All told, what we've been trying to do is leverage our donors' money and bring in grant funding to do this research. And all told, we spent about $250,000 so far just on Fusarium wilt to try and help you manage it and figure out ways to avoid it. So some of our literature's in the back. I forgot to bring my little stash up here, but we have some literature in the back, including some information about our website. If you go to our website, it's got all of the presentations from the seminars we've had, the videos, the results from the trials, all that stuff's on our website. So I do encourage you, if you need any of that information, to peruse our website. But also I want to mention that we work pretty closely with Western Growers, and Hank Gickless is here from Western Growers. They have a Center for Innovation and Technology over in Salinas, and we work closely together just to try to move at a faster pace in ag innovation. Hank is the one that introduced us to Pornima and Trace Genomics that you're going to hear from today. And in our trials, just to start the introduction to what today is about, in our trials, we actually sent soil samples to Trace Genomics to try to start calibrating. You'll hear about what they do, and the hope was that we can start getting calibration of a test that you could use to know whether you're likely to get pressure from Fusarium or not in a field before you decide to plant there. So we've worked with them with soil samples from our trials that we've done, and we've put in for some funding to figure out, because for this tool to be really useful for you, you need to be able to know what kind of sampling you need to do, right? Do you need to do one per field, one per acre, what kind of, so we need to learn the distribution of the disease and the protocol for sampling. So that's another area that we're hoping to do some research on with Trace Genomics. So anyways, I will stop talking about that. And I wanted to introduce Hank Gickless. Hank is the Senior Vice President at Western Growers for Science and Technology and Innovation, if I got that about right. And I appreciate him coming out today and working with us on all these things. And he's going to introduce today's speaker. Thank you, Hank.
Thanks, Paul. So I appreciate the opportunity, and I really appreciate the opportunity to work with the Yuma Center for Excellence in Desert Agriculture. Paul touched on it a little bit. I think we have common missions. We are both interested in solving problems faced by the fresh produce industry. For those of you that are not familiar with Western Growers, we are a trade association, and we represent growers and handlers of fresh fruits, nuts, and vegetables produced and sold from the states of California, Arizona, Colorado, and New Mexico. And just as a little bit of background, probably about 2.5 years ago, our board started talking about the fact that the industry faced a series of problems that likely would not be solved by any other means than accelerating, the availability of technology, to deal with some of these things. And it runs the gamut, everything from soil health, which I think you're going to talk a little bit about today, and disease prevention, things like that, to, automating operations to deal with labor shortages, to optimizing water use in the field, and the list kind of goes on and on. So we started this Center for Innovation and Technology in Yuma. Actually, Trace Genomics was one of our first resident companies there. They came to us in 2016, and I think you were founded in 2015. So, pretty young company. They've raised quite a bit of capital and have created a lot of excitement in both the tech community and the agricultural community. And I think we saw the promise of genetic sequencing technology early on when we first came across them pitching at a Thrive event, which is an accelerator program sort of in the Silicon Valley, Salinas area. Became interested in them, brought them out to Western Growers annual meeting where they won the Growers Choice Award. And I know that they're, affiliated with a couple of really, key accelerators, in the tech industry now, including the Illumina accelerator and the Terra accelerator, and Purnima and I have known each other since those first days in Thrive. Just a little bit about her. She's a 210 graduate of the Stanford University School of Medicine, and frankly, you meet the Trace Genomics team, and they're all doctors of one sort or another from Stanford University. It's very intimidating to be in their presence. She's done research with Nobel Prize-winning scientists. She's got 13 years of experience in genomics, specifically tracking infectious diseases, and has actually brought that expertise into the agricultural sector. And so with that as an introduction, I'll let her tell you about the work here in Yuma specifically and on Fusarium specifically, and I'll just reinforce our appreciation for the opportunity to work with the Yuma Center and with Paul and with Trace Genomics to try to solve problems for growers. Thanks.
Really excited to be here. What I wanted to do today was take you through the journey of trace, a little bit from the science and technology perspective, so you can see where the science and the technology is, how we are using it to solve problems, and then really delve into how we are using this technology to work closely with growers to help detect and predict Fusarium. I'm going to stand here because I'm going to advance my slides. Basically, the goal of our company is to provide the missing data layer in modern agriculture, which is a look into soil health and soil disease. Like, modern agriculture has a lot of tools available today, and we are accruing more and more tools. We're becoming very data rich. There is a lot of plant sensor data, there is a lot of weather data, moisture data, imagery, but really what is missing right now is the ability to measure soil health and soil disease. And so that is the layer that we're providing with a single genetic test. So we use genetics, and we use one test to quantitate both soil health metrics and soil disease metrics. And the reason, when we talk about soil is almost a universal input in agriculture these days, and there are different ways in which you can look at what happens in the soil that impacts crop yield, that impacts quality, that impacts shelf life, and some of these are soil type. We talk a lot about soil type and how there are different physical properties of soil types that really matter in terms of what you plant, where you plant, how you treat the soil. Of course, the nutrients, chemical testing is already being done today in many labs across the country, where you measure macronutrients like the nitrogen, phosphorus, potassium, calcium, also the microbes. You're measuring a lot of the smaller, the more minute elements in the soil as well. The other aspect of soil that's really important for yield is carbon and energy, and the one thing that trace really focuses on is measuring the bugs, the microbes in the soil that are really responsible for figuring out how the yield is impacted both by disease as well as by health. So what are these bugs doing in the soil? Every teaspoon of soil contains many millions of microbes or bugs. And all of these microbes work together to impact prediction of yield, to impact how much you get out of an acre of earth, what the quality of that produce is. And some of these microbes are harmful. They cause diseases like Fusarium, Burt, Sclero, Phytophthora, Pythium, all of those horrible diseases that you hear about that are soil-borne. There is a category of these microbes that cause those diseases. But not all microbes are bad for the plant. There are a lot of microbes that are also beneficial. And so these microbes are the ones that are helping nutrients cycle in the soil and make it available to plants. There are microbes that make nitrogen available to plants, phosphorus, potassium, carbon. There are microbes that really impact, for example, what is the health of a plant. There is like beneficial microbes in the soil. There's a whole class of microbes called mycorrhizal fungi. I'm sure a lot of you have heard about them. There's also plant growth beneficial bacteria that live in the soil as well, that all increase and boost yield and plant quality out in the field. And so when we launched Trace, our one goal was to be able to provide one test that measures soil health and disease and really aligns the insights that we're providing from this test with what are the critical input decisions that growers need to be making out in the field. If you have diseases out in the field, they really do impact your choice of varietals. They impact your choice of fungicides, other chemicals. If you have poor drainage capacity, for example, it really impacts the way in which you manage your lands. And also there's a new class of fertilizers, the biofertilizers that are coming out, that are becoming available to growers, that are all tied in with the beneficial bugs, the beneficial microbes that are found in the soil. And so the genetic tests that we do, we have actually brought a lot of learnings from the human space. So there are genetic tests in the human space that can capture entire populations, for example, of the microbes in your gut. So you have millions of bacterial cells in your gut that all function to make you healthy, that all help you digest food. And of course, if it gets off balance, it also causes diseases in humans. So these tests, they really leverage what is called digital fingerprinting. So you basically take a sample, you extract the genetic material of these microbes and bugs. So that's the second step, which is extracting a fingerprint of the microbes that's present in the sample. And then we have these new age machines called sequencers that can read that fingerprint, and then you have software that comes in and quantitates these bugs. First of all, identifies which ones cause disease, which ones are beneficial, but also quantitates them, how much of each of these disease-causing organisms as well as the beneficial microbes are present in each sample. And so that's basically what the trace genomics test does. It's one test that gets applied to every soil sample that comes in through our doors, and we capture the entire fingerprint of the microbes in that soil sample, both from the good and the bad microbes. So I pulled this pie chart from a real soil sample that came in through our lab. And in this pie chart, every slice represents a different microbe. And so you can see straight away that one, there are different quantities of different microbes, and two, there are many, many different microbes in every soil sample. And so, from our experience, every soil sample that comes in through our door contains tens of thousands to hundreds of thousands of microbes. And when we are capturing a digital fingerprint, we're obviously capturing the fingerprint of all of these tens of thousands to hundreds of thousands of microbes in every sample. And this generates a lot of data, a lot of information. And so what we have been doing at Trace, along with the community, is really understanding, first of all, what microbe is there in the soil sample, identifying those microbes, but also quantitating the microbes. So you need software to be able to do that. You need data analytics to be able to interpret what all of this pie chart and these rainbow colors really mean for yield and for disease from that particular grower field. And so we're also working very closely with growers as well as the community to use this information to inform those critical input decisions, those critical input choices around varietals, around product choices, as well as the drainage and fertilizers that I talked about earlier. How are we interpreting the data? So, when we look at every soil sample and those hundreds of thousands of microbes, the interpretation of those microbial signature happens at two levels. One is being extremely specific. So there are extremely specific microbes in that soil sample, like Verticillium dahliae, like Phytophthora, like Pythium, Sclerotinia. There's Fusarium oxysporum specific to lettuce. So these are very specific microbes in the soil sample that we can identify using software from every soil sample. And not only that, we're able to quantitate those microbes. So it's not just a yes or no, it's also how much of that microbe is present in that soil sample. And that's really what makes this technology powerful, any genetic technology, really powerful to quantitate risk on a field, is because it does allow you to quantitate these microbes. And the other part of it, the test is a lot more broader. It actually can generate a lot of soil health indicators. So what does soil health indicator mean? We go to a doctor's office, we get checked up for vital signs every time we go to the doctor's office, right? We get checked up for our blood pressure, our height, our weight. There's nothing similar that's done for soil. And so from a biological standpoint. And so that's what these soil health indicators are. We've been working very closely with scientists, both in-house as well as externally, to really convert all of this immense data that we're generating per sample into more quantitative metrics around nutrient availability, ecological indicators, and beneficials. And what do I mean by that? This is what I mean. So the nutrient availability metrics are measuring microbes that can cycle or solubilize the fertilizers that you're putting in, the NPK and the carbon sources. The ecological indicators include water use efficiency. How much drainage is there in your soil? Is there a waterlogging problem? We can actually figure that out from our test because we're measuring microbes that thrive under drainage issue conditions. So if you're measuring those microbes, you can measure water use efficiency. And then these are the beneficial organisms I already talked about. And of course, our test, the first thing that it spits out is a pathogen panel screen. It is a new technology, and so one of the things that we do quite extensively in-house, working both in-house as well as in partners with both YCEDA as well as Western Growers, is making sure that we are validating our test. And so we've done this for Fusarium oxysporum for lettuce. We've done this for all of the other pathogens that we are offering on our disease panel. We look at how our test performs with external lab tests as well as other internal tests that we do. When you add more of that disease-causing bug into the soil, our test detects more pathogen in that soil. So we've been doing a lot of these validation tests internally and externally. So now I'm going to shift and talk very specifically about how we're using this to detect Fusarium oxysporum. Fusarium oxysporum, a large part of that population is actually beneficial to plants. There is a huge proportion of Fusarium oxysporum in soil that's beneficial. Only a small number of species and strains actually cause disease in plants. And different strains cause diseases in different crops. So there is a Fusarium oxysporum that specifically had decimated banana in the 1950s. The banana that my parents used to eat no longer exists because this Fusarium oxysporum, this particular strain that's specific to banana, totally decimated the Cavendish banana, and now there's a new banana population that we're eating right now. Lettuce is Fusarium oxysporum latucae. Strawberry is Fusarium oxysporum frageriae, and then tomato you have a completely different Fusarium oxysporum strain. So the exact same Fusarium oxysporum, but different strains affect different crops and cause diseases in different crops. So if you think about a family tree, a family tree that each one of us, if you have your mother, father, children, who they marry, and so on and so forth, that's basically what this is for Fusarium oxysporum. It is showing you how related these different strains are, and this is just showing you the Fusarium oxysporum strains, and as you can tell, there are many, many, many different strains here. You don't have to read them, but this is just meant to show that this tree is very complicated, and that it's a very large family of pathogens which are very similar in their genetic content. And this is why diagnostics of Fusarium is extremely difficult. So for lettuce, it's like I mentioned before, it's a very specific strain of Fusarium oxysporum called Fusarium oxysporum latucae that infects lettuce, and that's melting all the lettuce out there. And so there are very specific methods like culture can detect whether there is Fusarium that's causing a plant to die, but it's not able to distinguish the Fusarium oxysporum latucae from the other strains, both the beneficial strains as well as all of the other strains that are infecting all of the other crops. So how do you know if you're having Fusarium oxysporum specific to lettuce, if it's a beneficial strain, or if it's one that's affecting strawberries and people in Yuma don't even need to worry about that? And so that's why we're using soil genetic sequencing. By looking at the genetic material of the Fusarium oxysporum, we can distinguish between both the beneficial and the pathogenic strains, as well as within the pathogen class, the disease-causing class, we can tell you which one is causing disease for strawberries, which one's causing disease for lettuce. We can pick up, identify, and quantitate these specific strains. This is real data from here in Yuma. And so what we did was we took soil samples from ranches 1, 2, 3, 4, 5, 6. These are 6 different ranches with different, with iceberg or with romaine lettuce, and some of them had different varieties too. The important point here is we had paired samples, healthy and diseased. These are soil samples around these plants. And so the healthy samples are basically soil samples from a region of, from an acreage that is healthy, has no Fusarium on it. And the diseased samples are basically soil samples from a section of the ranch that had Fusarium on it. And you can very clearly see with our test, we're not only picking up, this is like the levels of specifically of Fusarium oxysporum latucae, the one for lettuce. And so you can see over here that the quantity of this particular pathogen is different between the healthy and the diseased samples across these different ranches. This is in Yuma. And so what this really lends itself up to do is for us to be able to generate what are the community thresholds, what do they look like in a healthy soil. And so this is the community healthy average that we surface on every report that we give out to growers who take our test. And I just wanted to put this up there because I just wanted to show how this entire information gets translated and put in an easy to read report, because it is critical. You can have all the technology in the world, but if we are not translating it into a format that's readily readable, user-friendly, and actionable for growers, it's just not useful for the community. And so we've been working very closely with growers, with other folks in the community, to really come up with a reporting format that makes sense. And so this is what the report looks like. We bin pathogens by whether, diseases by whether you need to worry about them or whether you don't need to worry about them. So when a disease is found in your sample to be below the level of community average healthy, you don't need to worry about it because you're below what is the average that's found in healthy samples. The minute you're above that, that's when you need to start worrying because you're at higher risk. And so that's how we get these two categories. One is below the community value. All of these pathogens and diseases you don't need to worry about. You're not at high risk for them. Once you get above that community healthy threshold, that's when you start to get at higher risk. And this is also reflected in this data graph. The purple bar, so the bar to the very left, is the community value. And then there are two samples, and the pathogen abundance is the quantity, is also indicated for these two samples, RH and RU. And we do this for different pathogens, and this is done in a crop-specific manner. This is particularly for lettuce, but we also do this for strawberries, vineyards, nuts, corn, soy, other crops. So there are different ways in which you can use this test. We are a third-party testing platform, so what it means is that we're not providing specific recommendations for specific varietals or specific fungicides, but we make it really easy to test your field before you go into plant. And so that's the first use case, is to test soil prior to planting. Identify what the disease pressure is on every management block, and we can talk a little bit more about resolution in the next slide. Or some growers prefer to test at-risk blocks, ones that they know are at higher risk. And then obviously the reason why a lot of our growers do this is so that you can strategize your operations based on quantitative soil data. So you're actually measuring what's in the soil, understanding the risk before you go in and plant. The other test case, which is particularly relevant for Fusarium because we don't have a really good product out there that kills Fusarium, that allows you to manage Fusarium. There are some varietals, and of course YCEDA is doing a lot of field trials in this respect too, where there are some varietals that are more resistant to Fusarium. But if you want to be able to evaluate specific products and management practice efficacies on your own fields, some of the growers we work with, have their own suite of tools that they're testing, which is very confidential to that particular grower. And so again, we are bringing this tool directly to growers so they can assess for themselves what products and what varietals are really working out in their fields. A little bit about resolution. The note here, to come back to what Paul said, we haven't identified exactly what resolution of testing needs to be done. But one way in which we advise growers to do testing is to really test at the level in which they're making management decisions. So what this means is if you're making decisions on a 40-acre block, then you should be testing at that 40-acre block resolution. But of course, what that also means is you should be testing at a resolution that catches, because Fusarium is very patchy, you can certainly miss patches if you're not testing at a much lower resolution than your 40-acre block. And so a lot of growers tell us how, it starts off in 2 acres, and then the next planting season, it's basically gotten half the field. And so that's really why, 40-acre block, you could test, you can send one composite sample across that 40-acre block, but you will be missing those patchy areas like that 2-acre block that I said. So I almost, a lot of what the growers that we work with, we say, test on a per acre basis or a per 2-acre basis, depending on how much you're willing to spend on testing and also what results and insights the testing will provide to you. But obviously, this speaks for itself. The higher the resolution, the better you can identify what your risk is. I just wanted, that was my last slide. I just wanted to quickly say this is my team. There's two of my team who are here. Chris, who's at the back of the room, is our agronomist who's had many, many years of experience. And also Will, who's our customer engagement associate, is here as well. He's also at the back of the room. And so they're going to be around until about Thursday or so. And so I'll be around till tomorrow. If you have any questions, if you want to have meetings with us, please feel free to contact us. We'll be around. That'll stop. Take any questions. Yeah?
That's a good talk.
Thank you.
I'm just wanting to know, when we say percent abundance, is that in relation to other signatures? How many signatures do you see for percent abundance?
Sure. So the way in which we calculate percent abundance is across all fungi.
Okay.
Yeah, and so the test actually captures everything in the soil, and we bin the signatures by whether they're bacterial, or fungal, or pests, or nematodes, or others, including plant material. And so the reason why we really do focus just on the fungal population is because a lot of the competition happens within the fungal group in terms of like disease manifestation, and so we have done some amount of validation on that front. So our denominator for fungal pathogen is all fungi, for bacterial pathogen is all bacteria.
The signatures are specific or generic?
What do you mean by specific or generic?
You use.
Yeah.
Is it very specific to the pathogen, other species, or it is a kind of degenerate signature?
When we're identifying pathogens, they're very specific to that strain. They actually map to that particular genetic sequence. Yeah?
Could you talk a little bit about your collection procedure and how large a sample do you need for testing?
Sure. So the way in which we collect samples is very similar to how growers collect samples for chemistry testing. And the reason why we did that is because we just wanted to take one of the things out of the equation. So it's very similar to what they do. Either it's a composite sample or they actually send us 3 different cores. But that core has to be really mixed before you take a subsample and put in our soil preservation solution. So with soil biology, it is tricky to ship at a high temperature or with different humidity because the soil biology does change very rapidly. And so as part of our kit, we do ship out tubes with soil preservation solution in them. Yeah. The big advantage compared to traditional diagnostic techniques, the big advantage is that we're using one test for that entire pathogen screen. So it's not like we have a separate test for wort, separate for Sclerotinia, separate for Fusarium oxysporum. Each soil sample that comes in through our door goes through that entire pipeline. And at the end of the day, like depending on whether the grower wants a pathogen report for specific for lettuce, it's all software that basically says this is a list of pathogens that we have validated for lettuce, and so the report contains only those pathogens for lettuce. And if it's strawberries, it's a separate report, but at that point it's still, again, it's a software report, software generated report. Strawberry sample, a lettuce soil sample, vineyard soil sample, corn soil sample, all get processed the exact same way. And that's also why we're really able to standardize the way in which we're measuring these soil vital signs, the soil health metrics. We have just standardized that whole pipeline. So we are very quickly able to compare your sample to the community because all of the samples are treated the same way. Yeah.
Does it matter if the ground is wet, dry?
Yeah, so we typically suggest that soil samples get sent to us when the ground is not sopping wet. Also because it does affect the weight of the soil, how much soil you put into that soil preservation solution. And so typically we say, wait at least 24 hours before taking a soil sample after irrigation.
You would want to completely dry either.
I mean, it doesn't have to be what I meant to say, it doesn't have to be bone dry, but it should not be sopping wet. Yeah.
Are there different procedures for different soil types, or all soil types tested?
No, it's the same procedure for all soil types, and we have actually done a lot of validation on that end as well. And the reason why we kept it the same is because soil type is actually part of the data that comes in with every sample. And so as we're growing this community, the way in which we're looking at the community, that is definitely one variable that we will be able to to look at and to really tailor the community to. And so when I said, that community average was drawn only for Yuma, as we grow the number of samples that feed into that community database, we'll be able to say, this is the community specifically for this valley or this type of soil and so on and so forth. So it makes it a lot more precise. So soil type is something that goes into our into that database.
So you already have extensive data from various valleys here, or are you just getting started with that? Or?
So we have done one round of testing from the last planting season, and so now we're basically here to see how we can, help growers really increase that testing volume, and also to see how, if it is useful for growers to have that valley-specific community, then we're certainly happy to have that. No, we have actually not done those types of experiments in terms of figuring out under field conditions, because that's also driven a lot by temperature, is driven by what variety you have in the field, and is driven by a lot of other environmental conditions. So we have not done those types of field level experiments. Yeah. We're basically providing the very first step to understanding all of that, which is even figuring out if you have the bug in the soil, which is causing the disease, and quantitating that. Sure, so turnaround time right now is 3 weeks from sample receipt. And so that's why before planting is a really good time frame for a lot of the growers that we work with if you send us samples early enough before you make your planting schedules. And a lot of growers also send us samples, like I mentioned before, to test before and after treatments. And so under those contexts, it's all about understanding what products are working, what varietals are working out in the field, and really combining that timeline data with figuring out a strategy for the next planting season. Sure, the cost per sample is $199 for the pathogen panel, and that gives you information about all of those lettuce pathogens with one test. So it's actually priced very competitively if you think about how much just a single pathogen test costs out in the market. We also offer bulk discounts for over 96 samples submitted from the same customer. Yeah.
That's per acre?
That's per test.
Per test.
Per test. So if you test per acre, that'll be $199 per test.
And that's some of the research that we're trying to do now with them, is to figure out, to really get a good understanding of what the pressure is, what is the sampling protocol that you need to do. And obviously that'll impact the price, and that'll impact, whether it's something you want to do or not. So that's an area of research we're looking into now in cooperation with them.
Yeah.
Okay, if there's no further questions, we appreciate you guys coming. Like she said, they'll be around for a couple days. If you want to ask us how to get a hold of them, or you want to talk to her afterwards, you all have surveys in front of you. It would help us to help you better if you would fill those out and drop them in the back, just so we can respond to anything that would make these better. But in the meantime, thank you for coming. And again, thank you to those that support the center, and thank you to Western Growers, and certainly to Trace Genomics.